Hey everyone, it's Barbara again from the Kern Rivers chapter, and we are continuing the CIC Certified Infection Control Study course today, and we will be talking about topics in the lab to prepare you for the CBIC exam for Certified Infection Control. So we'll start off with immunology. And in immunology, we talk about antibodies and antigens. So antibodies are the good things that our bodies make to help us to fend off the antigen. In blood samples, we look for antibodies so that we can diagnose a variety of infections, specifically viral infections, because they're hard to culture. So there are many different types of antibodies, but specifically we'll be talking about the IgG and the IgM. The IgG antibodies relate to long-term immunity. So usually if you have an IgG positive, it represents past exposure to the disease. Whereas IgM antibodies are antibodies that are developed as the primary immune response, therefore they indicate current exposure to a pathogen and probably that you have the active infection currently. So the way of remembering IgG from IgM is if you have an IgM positive, you need to call your mom. So that's just a kind of silly way to remember the difference between IgG and IgM. Continuing in immunology to talk about titers. Now, titers are just so important for healthcare personnel to know what their titers are against different diseases. So a titer is a test that detects the presence and measures the amount of antibodies within a person's blood. So you draw somebody's blood and then the lab person will continue to dilute that blood into a test tube until there is no longer a positive reading. So it will always be expressed as a ratio. So here's what's happening in the lab. So you take a milliliter and you make a 1 to 10 dilution and you put it into some anti-serum and you keep diluting that until there's no more antibodies that can be detected, and that is your titer reading. So the higher the second number, like in this case 40, the higher that number is, the more antibodies are present and it indicates more of an active infection. Also in immunology, we talk about immunoglobulins. So immunoglobulins are special concentrated antibody preparations that provide immediate short-term protection against disease. So an immunoglobulin, the person that is receiving it, their immune system doesn't have to act. So the antibodies are already prepared for them so that they can get the immunity without a stimulation of their own immune system. So immunoglobulins are available for hepatitis A and B, measles, polio, rubella, rabies, and varicella. And may be appropriate to give people as they have been exposed to various diseases, you give them the immunoglobulin to help them not come down with the disease. So in immunology, we talk about different types of cells. For example, the white blood cells, which are really the warriors of the body. They account for only 1% of the blood cells, and they are either granulocytes or agranulocytes. So here we'll talk about the granulocytes, and there are 3 different types of those: the neutrophils, eosinophils, and basophils. The neutrophils are the primary defense against infections. Most of the white blood cells formed by stem cells in the bone marrow, they are short-lived, but you'll see an increase in neutrophils during an active infection. The eosinophils participate in allergic reactions and blood parasite infestations, and so if you see an increase in eosinophils, it could be due to those causes. And lastly, the basophils also participate in allergic reactions. They do contain histamine and heparin to help the body with those reactions as well. So a second type is the agranulocytes, which are one-lobed and they have no granules. And there are two types of agranulocytes, the lymphocytes and the monocytes. The lymphocytes are the main type of cell found in the lymph, and we have both T cells and B cells. T cells activate the body's immune response, and without them, the body is not able to elicit any type of immune response. And we have both CD4 and CD8 T cells. We also have B cells. B cells produce antibodies in the humoral immune response. And then our monocytes. Monocytes are very interesting cells as they are able to differentiate into a macrophage at the site of infection, and then these macrophages are capable of engaging in phagocytosis, which is the engulfing or the eating of another cell. So very fascinating cell here, the monocyte. We do have various barriers to infection, why we're not always walking around sick. We have the skin as a primary barrier to infection. We do have our mucous membranes and also the cilia in our lungs. These mechanical barriers to infection help to prevent the pathogen from ever entering the body. There are different types of immunocompromised patients that we are likely to see in our patient care settings. Those with leukemia or lymphoma, those that have AIDS, which is a CD4 T cell count of less than 200, those that are post bone marrow transplant. So for 1 year post bone marrow transplant, you are considered immunocompromised. Those that suffer the effects of malnutrition, we are coming across this more and more in our acute care settings and outpatient clinics. So we have an increase in drug abuse and also in poverty. And then the last type of immunocompromised, when we have neutropenia, which is an absolute neutrophil count of less than 500. So different types of immunocompromised patients that we are likely to see. Vaccines that are recommended for healthcare personnel, of course, influenza. We all know how important that is. Hepatitis B, the vaccine is 3 doses, and it is an OSHA mandate that this be provided to all healthcare providers. Measles, mumps, and rubella. We have an increase in measles going on right now in the country, and so it is important to make sure that we are vaccinated as healthcare providers against that. And then varicella, of course, and tetanus, diphtheria, and pertussis, as we know, also is very important for healthcare personnel. So some laboratory topics here. We are going to first talk about specimen transport, and this is a general rule that specimens should be transported within 2 hours of collection and kept in room temperature in a leak-proof specimen containers. It should be sealable in a leak-proof bag. Blood culture contamination rate should be kept at less than 3%, and if your lab isn't monitoring it, then as the infection preventionist, you should be able to speak to what the blood culture contamination rate is. When we talk about controls in a laboratory setting, we either talk about positive or negative controls. The positive control should grow out or produce a response. So if you're growing a bacterial culture and you do a positive culture, you'll actually inoculate your medium, whatever you're using, with bacteria and make sure that it grows. And then a negative culture is quite the opposite. You would expect nothing to grow in that. So sometimes you do a negative culture, again, in a microbiology lab. You just pour some agar on a petri dish just to make sure that your agar and your petri dish aren't contaminated and you're getting growth from that. So positive and negative controls. Antimicrobial resistance. So a few topics in the antimicrobial resistance. We talked last video about the role of the plasmid in participating in antimicrobial resistance. What's occurred in the laboratory, they use what's called a MIC, or a minimum inhibitory concentration, to describe the lowest concentration of a drug that can be used that is still effective against the pathogen. A key pathogen that we see that has antimicrobial resistance is ESBL, extended spectrum beta-lactamase. It is resistant to cephalosporins and sensitive to ciprofloxacin. One of the emerging multi-drug resistant organisms we're seeing a lot more of, unfortunately, is CRE, carbapenem resistant Enterobacteria acei, is resistant to carbapenems and tests positive with the modified Hodge test. There's also like the Carba-NP test that some labs use. It just depends on what test your lab is using, but it is looking for this enzyme to be resistant to the carbapenems like imipenem, meropenem, doripenem, ertapenem. Different laboratory tests that we need to know about for the exam. The Kirby-Bauer test, which frankly I've always found fun to do. So microbiologists, you plate just a petri dish of bacteria and you see what grows. So you put these discs on this field of bacteria, and each disc contains a different type of antimicrobial, and then you see which one is effective against killing the bacteria. In this case, in the slide, every disc of antimicrobial is effective against this particular bacteria, which is what you like to see. Also in the laboratory, they use a pulse-filled gel electrophoresis. This is when DNA is extracted from an organism to obtain a chromosomal pattern, and this is then used to assess the degree at which different isolates are related. They use this, of course, in looking at outbreaks. Then the PCR, we hear of PCR quite a bit in our field, and you'll hear various tests are done by PCR. Well, what that means is it's a polymerase chain reaction, and it's a type of molecular testing where DNA is amplified. So you take some DNA from the sample, stool sample, blood, whatever, and then you put it into the PCR test, and you take just a little bit of DNA in that sample and you get a whole bunch of DNA. So that's what it means. It is amplified. You take a little bit, and at the end of the test you get a lot. The Will-Felix agglutination is a test performed to differentiate the rickettsial antibodies. We'll talk more about the rickettsial diseases in our next lecture on diseases. But rickettsial diseases like Rocky Mountain spotted fever, which is transmitted by ticks. And there's the slight agglutination test that you can see will either be positive or negative based on the amount of clumping, which is what agglutination is, that is demonstrated on the slide. So in the laboratory, we use a variety of culture media. Media is used to grow a variety of microorganisms, and there are different types, such as nutrient agar. Nutrient agar supports the growth of a large different amount of bacteria. So if you just want to see what's in your sample, you use a nutrient auger, and it's going to support the growth of a large number of different organisms. An enrichment medium has specific nutrients for hard to grow organisms, which are also called fastidious. Selective media selects out certain commensals by the addition of inhibitory compounds. So for example, maybe you're trying to grow a yeast in a culture medium. You might want to add an antibiotic. So that's going to eliminate the growing of commensals. The commensal bacteria will be inhibited by the antibiotic, and you'll be allowed to grow your yeast. Differential media allows some organisms to grow while inhibiting others. Sometimes we're going to look for different anaerobic bacteria. Remember, those are the bacteria that do not require oxygen. So these bacteria are common in such specimens as those from the rectum, the nasal passages, the sputum, the urethra, or voided urine. And so it would not be appropriate to culture for anaerobes in specimens from these locations. So we're at the end of this section on laboratory and immunological basics. We will recommend now that you go to these chapters and exams and you look up the questions after studying the material. And again, the idea is after you have studied all this material, you will be able to answer the different questions that are outlined here. Good luck in studying, everybody.